Gauss on this side. So today hand we are dealing with a topic called as gene interaction by means of epistasis. So let's get started. First thing first, we should know the definition of epistasis. So basically, epistasis is a type of gene interaction in which one gene masks or suppresses the action of the other gene. Okay, deal with. So basically, epistasis has a lot of different types, but still today hand we'll be dealing with the first two types of the epistasis, that is called as recessive epistasis and dominant epistasis. So let's get started. Starting with the first thing first, called as recessive epistasis. So here the ratio of the recessive epistasis is 9:3:4. Okay, so whenever the recessive alleles at one locus are masking the expression of the other alleles at other locus, it is known as recessive epistasis. Okay, deal with. Again, when the recessive alleles at one locus are masking the expression of other alleles, like regardless of their dominancy or the recession, that epistasis will be called as recessive epistasis. Okay, now this type of genetic interaction is known as supplementary epistasis as well. This type of genetic interaction is also known as supplementary epistasis. And basically, if I'll be dealing with the best examples of the epistasis, here it is the grain color in maize. It is the grain color in maize. Basically, there are three colors of the grains. They are called as purple, red and the white. Purple, red and the white. So how come the purple color is developed? So because of the capital R and the capital P, whenever the genotype is having capital R and the capital P both, then only the purple color is developed. Then only the purple color is developed. Whenever only capital R is present, then the red color will develop. And whenever small r and the small p are present, then only recessive white color will develop. So for example, whenever we will be dealing with the cross between capital R, capital P along with the small r, small p, what colors will obtain? So here it is the homogeneous purple color, whereas here you are having double recessive white color. Do remember one thing, that in a normal gene interaction, dihybrid interaction, these two genes will be having different actions, but in case of the epistasis, two genes are governing the same action. Okay, deal with. So we are crossing purple with the white. So here the example is in front of us. First of all, the parents. So here the parents are the purple and the white pure, which means capital P, capital R and the capital small p, small r. Okay. Now their first filial generation will be called as F1, which will be the hybrid of both of them, capital P, capital R and the small r. And the phenotype will be related with the dominant one only. So it will be purple colored grains. Okay. Now here, in case of the F2, we will be having 16 different results because of the Punnett square. So the gametes have obtained by the selfing of the capital P, capital R, small r along with the capital P, capital R and the small r, and we will be having these four gametes. Capital P, capital R, capital P, small r, small p, capital R, small p, small r. And again here capital P, capital R, small p, small r, small p, capital R, small p, r and the small p, small r. And the results here will be, the first one will be having capital P, capital R. Okay. Then the other one will be a capital P, capital R, small r, capital P, small b, capital R, small r, then capital P, capital R, small r again. Okay. So deal with these cases as well. So whenever we will be going for all of them, we will be observing that they should have one capital P and one capital R so as to have purple color. So if I am going for like those capital P and the capital R here, like the second, the third and the fourth. So how come, if I will be checking them out, how many of them are there? Yes. Okay. I call this as a triangle. I call them as a triangular formula because ultimately if I am dealing with them, you can observe that these 9 are having capital P, at least one capital P and at least one capital R in their genotype. So here you will be having 9 purple. Here you will be having 9 purple. But if you are checking about the red as well, you will be observing that the small p should be there. The small p should be there. So here you are having red, this one is also red, and this one is also red. Okay, so 3 red you are having. And then if you are dealing with the white, so you will be observing at least one capital P should be there along with the small r. So here 1, 2, 3 and 4. This is what I call as the recessive epistasis in the maize grain color. Okay, and the normal 9:3:3:1 has been modified into 9:3:4, which means last 3:1 has been merged into each other, and now a new genotype is coming. Here you will be having only 3 phenotype, that will be the red, purple and the white. The red, purple and the white, which will be 9 purple, 3 red and 4 white grains. Okay, deal with. Now dealing with the next topic. Here, epistasis gene interaction in the dominant epistasis. Epistasis gene interaction in the dominant epistasis, which will be having the ratio as 12:3:1. What happens in the dominant epistasis? When a dominant allele can mask the expression of other dominant and the recessive alleles at another locus. What does locus means? So locus means it is the location of a gene or allele at a specific DNA segment. Okay, deal with. Now the example of the dominant epistasis is found in the fruit color of summer squash. Fruit color of summer squash, or rather, like, the scientific name of the summer squash is none other than Cucurbita pepo. It is Cucurbita pepo. So deal with that as well. So here we have 3 major colors of the fruit in the Cucurbita pepo, rather like we call it as cucumber. They are white, yellow and green. They are white, yellow and green. So the white color is controlled by the dominant gene W, and the yellow color is by the dominant gene capital G. Okay, so basically white is dominant over both yellow as well and the green as well. And if I are dealing with the green, the all should be in the recessive condition. All should be in the recessive condition. So let's check it out. So first of all, we are dealing with the parents here. Okay, so the parents genotype is white. For white fruit you should have capital W and small g, or for yellow fruit you should have small w and the capital G. So the hybrid or the F1 will become none other than capital W small w and the capital G small g, or what we call as a phenotype. It will be matching with the dominant parent. It will be white fruit. Now whenever we are dealing with the small, like the F2 or the second generation, which means the selfing of the first generation, here you will be having capital. Okay, now here, like the capital W capital G, capital W small w capital G and the small w small g. The same case is here as well. Capital W capital G, capital W small w capital G and the small w small g. So the first one will be again capital W capital G capital G. The same case here as well, like the both, at least one should be dominant from both of them. So here like the same again, this as well, and this as well, and this as well. 9. But still we have another W's as well, because capital W is dominant over them. So here like these will be added in M as well. So now if you are counting them, you will be coming into conclusion that there are 12 white fruits. There are 12 white fruits because one gene is dominant. The first gene is dominant regardless of the dominancy or the recession of the other one. If only one single allele is in capital form, it will mask the action of the other alleles, and it will going for the formation of the white color. You can observe in the third row second column, or the second row third column. Okay, second row third column as well, and the fourth row, the fourth row and the second column. Okay, only one single W is present, but still it has masked all the expressions of the other alleles. And in case of the second one, if you are dealing with the capital Y, so here like there should be like the small w capital G, small w capital G small d, or the small w, capital G, small g. Then only you will be having Y color or the yellow color. And at the end of the day, if you are having small w and the small g, then only you will be having the green color. So here the new ratio will be coming. Here the new ratio will be coming in front of you. That will be called as a 12:3:1. That will be called as 12:3:1. So deal with these cases as well. Okay. Now these were the two types of the dominant epistasis and the recessive epistasis, in which one of the gene was masking the effect of the other one. We will be continuing this portion, but still, like if you are having any kind of doubt, you can ask me there in the comment section as well. Okay, done. Thank you very much for your so much support, guys. Thank you.